So I should explain that the purpose of this discussion is to highlight issues around commercialization of cell therapies and challenges that we face in getting potentially useful products into the clinic. And this discussion is being filmed and will be available, I suspect, through the UKRMP website. So in the interest of time, we'll probably keep the discussion quite short. And I'm going to start off by introducing the panelists and telling you a little bit about what their particular areas of expertise are. So starting off with Dave Mooney.
Yes, good afternoon. Dave Mooney from Harvard University and the Wyss Institute, and my interests mainly are in the area of utilizing materials to regulate cell behavior.
Thank you. And I can't pronounce your first name.
That is a real challenge. So my name is Eleuterio Lombardo. I am scientific director at TiGenix. My responsibility is the basic research and the preclinical studies in the company, working with adipose stem cells and recently with cardiac stem cells.
Hi, I'm Stuart Forbes. I'm a researcher in the area of liver regeneration and development of cell therapies.
I'm Cathy Prescott. I'm an independent consultant for the company Biolatris. And for the purpose of this afternoon, my interest really is in around how we're going to solve getting these products, once they have been approved, onto the market, how we're going to get them paid for.
Thank you very much. So one of the issues which we're not going to discuss is the complexities of getting regulatory approval for a potential cell therapy. And the reason that we are not going to discuss that is that, of course, the rules are very different in different countries and for different products. We've also heard that there's quite a lot of funding in the academic sector for the early phases of development of a potential therapy and getting into phase 1 trials. So what I would like to discuss with the panel is at what point does or should academia start discussing with the commercial sector, and what do you see from your own experiences as the opportunities for success there? So Dave, maybe I should start with you.
Okay, so I will not try to address all that question, but maybe just a couple points. One is that I think it's very useful to very early on begin a discussion in terms of commercialization. However, I would also indicate that that does not mean to have a deal or to hand off the technology at an early stage. I think early discussion to get feedback for what the path might need to look like eventually to commercialization is very important. But I do think it's very important to also maintain some ability to run the early trials in the scientifically sound way that is not driven solely by commercial interests.
And so is it important to have all of your patents filed before you have those early discussions, or should you discuss them and then use those discussions to formulate the intellectual property protection?
So I would be a proponent of having your IP filed before one begins to have those types of conversations.
And so maybe, Stuart, I could ask you about the IP landscape around a purely cellular product.
Well, I think I don't think there's one answer. I think there are some products where I think there's IP on the cell itself, if I may say so, or the derivation of a cell, and others where it may be just about some manufacturing aspect of it. So I think the degree of IP may change during the clinical development of a product.
And so from an industry perspective,
yeah, I totally agree. Sometimes we are, as a company, approached by academic institutions too early. I think it has to be early, but we need some mature data to start talking. So that, you know, not that with some in vitro data, we need to have at least a good efficiency, for instance, in vivo animal model, something like that, or even already with some kind of phase 1 data obtained. But that would mean that we would kind of license a product or something that is already under development. If we want to help in the development, we really need to be involved at the early beginning, but we need some kind of mature data. Regarding patentability, we were talking before, I think patents are important, but sometimes we focus too much on patentability. And sometimes it's very important just to keep the knowledge. And that can be even more interesting than just patenting. You are patenting, it is public, it's nice, but it's public. So everyone will know how you do things. And at some point, it might be actually tricky to know whether this company is doing things using your patent or not, because at the end, we are using, all of us, stem cells that you can just take from a tissue.
And one topic that definitely raises the temperature is the role of academic tech transfer offices, whether they are helpful or an impediment to what you're trying to do. So any comments on that? Maybe Dave?
Be nice. Yes, I think they can be both. I think that as academicians, our job is not to go out and find the right opportunities and spend lots of time dating potential commercial partners, and that is a very time-intensive process. So I think it's not something that most of us have the time or the inclination to do well. However, the downside can be that sometimes those offices can start to put too high of a value on, particularly some of the early technologies, and that can hinder the progression and the partnerships.
I might do a little plug in the UK for this cell therapy Catapult, because I think a number of academics have found it useful to go into a meeting with them. It's a bit like a Dragon's Den competition, and just really think out loud about the possibilities and get advice about how best to approach the university tech transfer sector, potential partners, potential sources of funding. But I do think it would be true to say that many academics are feel that relationship with their university tech transfer office can sometimes be problematical.
Can I make a comment? Because as a consultant, I often work with technology transfer organizations. And in my experience, they are on the whole, pretty open to taking on board advice and thinking about really where are they in terms of the value of the opportunity. I also, I'm based in Cambridge and happen to sit on the translational advisory group for Babraham. And among that group, I think without exception, these are representatives from industry as well as from the Cambridge University Technology Transfer Organisation. And they will volunteer their time to sit and work with academics to help support thinking about what are all the issues that you're going to need to address when you're pulling that opportunity together in order to apply for further translational funding.
Thank you. So the next question that I wanted to ask was how long it takes to go from proof of concept to putting together an application for funding for a clinical trial. So maybe, Stuart, we could start with you, because you presented your clinical trial that is funded for macrophages in the liver.
Well, I could say 4 years, but it depends where you start measuring. So, I mean, we've been looking at the area, for example, the macrophages for a number of years, and it's matured, and then I guess we had some cell therapy, if you like, experiments in mice, which were very positive. So then we started thinking, well, maybe there is a possibility, and started looking at human cells, and it also looked encouraging. Then we started looking at scale-up. And what was pivotal was very early engagement with SNBTS, the Scottish National Blood Transfusion Service, who have experience in developing novel cell therapies, but also with the cell therapy Catapult. And what we found is that really we saw eye to eye that we wanted quite a low-tech, low-cost solution that may even fit within a national health framework. And that enabled us to get funding from the DPFS panel, who were also of that mind. So it depends where you measure it from, but it can take years. Equally, we've found with some data I didn't present on CSF1, we found that CSF1 predicts survival or death if you get fulminant liver failure from paracetamol. We were able to, through collaboration, to use the CSF1 stabilized compound and show efficacy in our mouse models. And very quickly, we're now looking to do a GMP manufacturing. So that's been really within 2 years. So it can happen very rapidly if it's a pharmaceutical or molecular approach that involves the cell rather than direct cell manufacture, which I think is inherently slightly slower.
So I think that's a good point that we should turn over to Dave, because you have a tremendous track record in developing really interesting biomaterials. So if from a sort of acellular perspective of regenerative medicine, what's the time frame?
So I think there's not a time frame, but in general, it can go quicker than a cell-based therapy. I guess my general thoughts on this would be that you really benefit, or really balancing the benefit and the potential risk. So if the benefit is potentially quite high and the potential risk of that therapeutic is low, so for example, you're taking a material that is well established and let's say a protein that already has a good track record of use in the clinic, and you're applying it to a patient population that has very little to lose, your ability to go quickly to the clinic is very high. On the other hand, if either the biologic has significant risk or these patient populations don't have that much to gain, there's already an existing therapy that is somewhat effective, then that changes the ratio tremendously. So, cell-free approaches can go quickly, but you still have to balance, risk and benefit.
And from a commercial perspective, how do you approach clinical trials?
Well, I totally agree with them. It may depend. Just as an example, we recently not started, but we submitted to the ethics committees and regulatory agencies the protocol for a clinical trial on sepsis. And our experience on that has been, I would say, like 1 year of screening all kind of inflammatory indications that could fit with the mechanism of action of mesenchymal stem cells. Then we selected like 3 indications that we identified that there would be an unmet medical need, there would be a market, cells could potentially work, and so on. And finally, we decided to go for sepsis. And that was 2 years ago, and it took 1 year to prepare what we think is a good protocol for a clinical trial. So this is a particular situation, maybe fitting with what you said, that there is an indication with a clear medical need, and we went really relatively quickly. So about 2 years to decide the indication and go for a phase 1/2 clinical trial.
Okay. I'd now like to ask Cathy to lead the discussion about costs, the costs of delivery, how you factor that into planning for something that you hope will be efficacious.
So cost, of course, who's going to pay for these products? As we know that many of these cell therapies are going to be potentially relatively expensive by comparison to conventional therapeutics, and that reflects, of course, how the complexity of the manufacturing process. In understanding how well do these products work, for many of these products we're looking at accruing benefits over years in some instances. And so in fact the whole question around reimbursement, if these products are really safe and they really work and they work as well as we think they will, then the whole question of reimbursement actually isn't the issue. The question is cash flow. And it's cash flow from the perspective of the company developing the product, because as soon as they get it out onto the market, they want to have payment for it because they've had investment in R&D and so forth. From the payer perspective, so the NHS or the private payer, from their perspective, what they're looking at is actually what's the risk that it works for the first year, but maybe it doesn't work quite as well for the second year, and perhaps doesn't work by year 3. And yet you're asking them to pay, potentially upfront, for something which you're saying is going to work over the next let's say, 5-10 years. So it's about how do you manage the risk relative to the cash flow, and that's really where we need to start thinking about developing different types of payment models. And there are some interesting models that are coming out that the NHS in particular is really driving. And that's around, for example, actually handing the risk back to the developer, saying, we, the healthcare provider, we will pay for the therapeutic, but if after time the therapeutic doesn't continue to work, you, the developer, must basically reimburse the insurer. This is kind of interesting because for some of these products, and TiGenix, I think, is a nice example with ContraSelect, you need the patient to also have a rehabilitation program. And you, as the developer, don't necessarily have the infrastructure or the ability to ensure that the patient is being compliant with the rehabilitation program along with the therapeutic. And so if a therapeutic doesn't continue to work, it may be because of other factors. So it's a really complicated picture that we need to really get to grips with.
Maybe, Stuart, staying with the subject of cost, people know in the UK how much it costs to transplant a liver and maintain a patient on immunosuppressive drugs. So with your NHS hat on, what's your feeling about cost for these new therapies?
Well, I think if you can produce a treatment which is of relatively low cost and is highly efficacious, the uptake is likely to be very high. If you produce a very expensive treatment, you're likely to have a niche market within the UK. There may be the potential to set up centres for clinical regenerative medicine where you could target the rich, but I think but most of us are motivated to try and get products that are affordable to society as a whole. So that's certainly something that we're interested in. So we have quite well set out costing brackets called quality adjusted life years, and those are something that we can measure potential efficacy against, and that's useful for us to judge efficacy versus cost.
But I think that the challenge is actually to have a high efficiency product, which is, I mean, if you have that, there will be no problem for reimbursement. But unfortunately, so far, we don't see that in the cell therapy industry field or landscape.
So that leads me on to my last question, which is I'd like to ask each of you for your idea about the future of cell-based therapies. So Dave, from a materials perspective, do you think we need cells?
I'm sure we will need cells at least some of the time, but and as I look to the future, what I see is a lot of the current work is going to establish proof of principles that cells can be therapeutic agents, but as time goes on, we will understand better and better why those cells provide therapeutic benefit, and we will replace those cells with either defined molecules or substrates of other types.
Thank you. The TiGenix perspective?
Of course, I have to say that there is a great future. This is going to be recorded, so I have to be careful with what I say. No, I think that there will be a future, but I think my feeling, I've been an academic scientist all my life until I joined the company. I have the feeling that we rushed in this field too much. So we all wanted to go to a clinical trial without really understanding what we had in our hands. And I have the feeling that, we see trends to efficacy, so on, but not strong data so far. I have to say that recently we finished a phase 3 clinical trial for the treatment of perianal fistulas in Crohn's patients, and we achieved the primary endpoint. So we are actually now working in the filing for marketing authorization with the EMA. So hopefully, if everything goes well, we will have a product, a cell therapy product, by the end of next year or something like that. But I think that right now what the field needs is someone, TiGenix or any other company, showing that cell therapy can work That would be very important. And then there are many scientific, actually basic questions that we need to understand, from who is the best donor, if we can identify the best donor for cell therapy, what is the patient population that should benefit, biomarker studies to define the patient population, the efficacy. There are a lot of work to do in order to really have such a great product that, of course, will be reimbursed if we are able to get it.
Final thought from you, Stuart?
Definitely need the right cells for efficacy. If tissue's damaged, you may well need artificial niches or molecules or matrices to help that tissue repair. And then if we do that, then we need properly powered trials. We have a great track record in that, and we should use those to tell us whether it works or not. Otherwise, the experiment is not helpful.
Thank you. Cathy, last word.
Last word. And so my thought is it's really about integrating with other disciplines, particularly around sensor technologies. At the end of the day, if we want to get these things reimbursed, we're going to be able to need to measure that they continue to work and are continuously safe over a long period of time, longer than our current trials. So as you move into sort of a phase 4 setting, particularly with the adaptive licensing approaches, we're really going to need technologies that allow us to measure, do these things still work?
Okay, with that, I'd like to end. The panelists will be around if you want to ask them questions individually, but thank you very much.